A very good morning, children. How are you all? Hope you understood the first session of diversity in living organisms. So now we come to the second session of the diversity in living organisms. In the last session, we have read about the different kinds of leaves and their shapes, length, okay, and the color and the margins and kind of venation they have. So, and all these are the things which we have read in the previous session. So now what we are going to see is we are going to check the observation of external features of the plants, external characters of the plants, especially the monocot plants and the dicot plants. Now what are these monocot plants and the dicot plants? We will study now. What you have to do is you have to collect around 5 different plants, especially make sure that there are maize, grass and paddy, wheat or these kind of plants in collect them from your surroundings and take them as the experimental material which are having the flowers. They should have the flowers. Okay, that is a point you have to keep in your mind. And then you make a small table, and in this table what you will do is you will make sure that you will fill all the columns of the table. So now I will tell you what are the things you have to check, what are the things you have to compare. Okay, now we'll go ahead with the table. Okay, so now what are the things you have to make sure you will find out from your experiment, right, from the activity. So what are the different things which you have to write it as a table. So the first one is, what is the name of the plant which you are choosing, right? What are the plant you choose and all, you write that particular name. Second one is the length of the stem, that is, so see that what is the size of the stem, so you measure it with a scale, or you can use some measuring tapes and all, then you can find out the length of the stem. Okay, that is what you can do. And then coming to the length between the nodes. Now what are nodes? Okay, if you see the plant and all, right, you will observe this kind of cuts in the plants. Okay, so okay, you can see this kind of cuts in the plants, and also these places, these cuts are called as the nodes. What they are called as? They are called as nodes. Okay, let me write it clearly to you. Nodes, right? Now the space between one node and the other node is called as the internode. It's called as the internode. Now see the size, okay, the length from one node to the other node. Okay, this is called as the nodal length. Okay, so what is the length between the nodes? That means you have to measure the distance between one node to the other node. So whatever the distance is, you can write it down. Okay, that is what you can do with the length between the nodes. And next one is the leaf venation. Already we have seen the two kinds of venation, right? One is the reticulate venation, next one is the parallel venation. So you can go ahead with the reticulate venation, or else you can go ahead with the parallel venation. So whatever the leaf it is and all, you can write the leaf, right? Next is the flower. Is the flower coming out as a single flower, or it is coming out in a group? You might have seen the different flowers too, right? They come, sometimes they come in the groups, or sometimes they only come in as a single flower. So you have to write whether it is a single flower or the flowers are coming in a group. Next one is number of sepals. Now what are sepals? Okay, now you might have seen a brinjal, right? So if you have seen a brinjal or a tomato, you can see, what you can observe here is, you can observe some green color. Okay, these kind of things. Okay, right. So these are the sepals. The green color which you can see here are called as the sepals. they actually hold the fruit, whether it is tomato, okay, whether it is tomato or whether it is brinjal, whether it is chili, whatever it is. So the green color endings here, they will hold the fruit from falling down. So these structures are called as the sepals, sepals, sepals. So you have to see how many sepals are there. Okay, how many sepals are there? And next one is the how many petals, right? So if you see the colored structures, okay, which are really very attractive and also which give out a green, which will give a very sweet fragrance, are called as the petals. They are called as the petals. So count the number of petals. How many petals are there in the different flowers or the different plants we have taken. Okay. And then coming to the taproot. Okay. So here there is a difference. Okay. There are two different kinds of roots. Here. One, you can go ahead with the taproot, right? So now let me draw the taproot here. I'll draw it for you. Now the taproot will have a main root like this. It will have a strong main root, okay, and then you can see some other very delicate, tender, okay, roots like this, okay. This kind of root system is called as taproot system, and this taproot system is located deep into the soil. You can find it very deep into the soil. Plucking of this kind of plants will be a bit tough. You need to put some extra energy. Okay, so this kind of system is called as the taproot system. So now in the same way, okay, now if you take the grass, and all, no, okay, so this is what you can see above the soil, and under that again you can find the hair-like structures. Okay, you can find the hair-like structures, and this kind of root system is called as fibrous root system. So you can find in every plant you can find two types of root systems. One is the fibrous root system and second one is the taproot system. So now you have to check whether this particular plant belongs to taproot or fibrous root, okay, or the plant which you have selected has taproot or the fibrous root. So that is how what you can do is you can easily fill the table. Okay, this is not a very hard or difficult task, so it's a very easy task. Again, you can do it very easily. So this is the one of the very important table by which you can know lot of differences. By this we can come to know that variety of characters present in the plants. Variety of characters are present in the plants, and you will come to know with this particular activity. Okay, children, so as an example, I give you a small table where we have taken some 5 plants and how they vary in their characters. So you can see on the screen. Okay, you can see on the screen. So there are 5 different plants which are being taken. Okay, so the first one is the guava, second one is the rose, third one is mango, and fourth one is paddy, and fifth one is maize. Okay, these are the 5 plants we have taken. Now you can see the stem, right? The stem of the guava plant is around 15 feet. The rose stem is only just 2 feet. Mango is 30 feet. Paddy is 2 feet. Maize is 6 feet. That means you can see lot of variation in the length of the stem, isn't it? So 15, 2, 30, 2, 6, right? Lot of variation in the stem. And second one is the length between the nodes, right? 5 centimeters, 3 centimeters, 10 centimeters, okay, and 15 centimeters, right? Here you can observe that, okay, here you can observe that even the length between the nodes also will differ, right? It's about 5, 3, 10, 15, isn't it? So the internodal distance or the length between the 2 nodes is also completely varies, right? It completely varies. And when you take the leaf, right, when you check the venation of the leaf and all, guava, rose and mango, these 3 have the reticulate venation. They have the reticulate venation. When you check the paddy and maize, they have the parallel venation. Now when you see flowers born in a group or born in single, guava, rose, mango, if you check these three flowers, they are born as a single flower. And when you see the paddy and maize, they are born as group. Okay, the flowers are born in a group. So that is how even the flowers, the way the plant gets the flowers also varies. And number of petals, if you see here, the first three, that is guava, rose and mango, have 5 petals each, and whereas paddy and maize, no, it's not. One thing I just want to make it clear to you, that number of petals here, 5 in the sense it doesn't have only 5 petals. Okay, one particular roll will have the 5 petals. Okay, one particular round will have the 5 petals, right? So these are called as the 5 petal flowers, and then there will be 3 petal flowers. So if you check the flowers and all, there will be 5 petal flowers and the 3 petal flowers. Okay, so here what you can check here is guava, rose and mango will have the 5 petals. They are 5 petal flowers, and paddy and maize will have the 3 petal flowers. Now coming to the number of sepals they have, all of the taken examples have exactly 5 sepals. So there is no variation seen in the sepals. And coming to the taproot or the fibrous root. So taproot, guava, rose and mango, you can see them as a taproot system, which are located deep into the ground, which have a very strong root system. But whereas paddy and maize, they are made up of fibrous root system, so they have the fibrous root system. Okay, so that is how you can see lot of variations in the 5 plants which we have taken as a example for our activity number 2. So that is how you can see the variations in the plants. Okay, now we will try to see one more example of a different set of flowers. Okay, now what we'll observe is the seeds, right? How different are the seeds? Can you see any kind of variations in the seeds? Before that, I would like to tell you what are monocots and what are dicots. So now we'll try to see what are monocots and dicots, and then we'll go to this next experiment. Okay, let's go. So now these monocots will have 1 cotyledon, dicots will have 2 cotyledons. Right, now coming to the what is cotyledon. These cotyledons are the embryonic leaves which you find after the germination of seed. So as soon as the seed germinates, what you can see here is you can see 2 embryonic leaves, right? So these are not the true leaves. These are not the true leaves, but they are the embryonic leaves which actually gives support to the plant, and also to support to the seed, and also support to the leaves, the true leaves which are going to come out till they start the process called as photosynthesis. So till the new leaves come out, till the true leaves come out, this particular plant requires what? Nutrients to survive. So these nutrients are actually given by these embryonic leaves. Okay, so now basing on these embryonic leaves, okay, the plants are divided into monocots and dicots here. So in the monocots, if you see the seed and all, no, like only one particular leaf comes out, that is the embryonic leaf. This is not the true leaf. I'm not talking about the true leaf, right? It is the embryonic leaf which actually comes after the germination of seed. Okay, so this particular leaf, it will stay till the leaves comes out from the plant. Okay, until then it gives the nutrients. Okay, this kind of leaves you can see in the maize plant. Coming to the dicots, you can see, okay, from the seed, 2 leaves comes out, right? So these are 2 embryonic leaves. Till the new leaves come out, till the original true leaves come out, these 2 leaves will give the or provide the nutrients to the plant. So that is how these two are the embryonic leaves. Because it has two, we call it as the dicotyledons. Here we have only one, so we call it as the monocotyledon. So even the leaves which comes out from the dicots and monocots are also different. They are not similar. Okay, if you see the monocots and all, no, right, as we have discussed here, these kind of leaves will have the what kind of venation? So when I'm drawing, you can easily guess, right? So they will have the parallel venation, and the dicots will have the reticulate venation. Okay, so and now you can see this is a bit broader in size leaf, and here this leaf is narrow and slender. Narrow, slender leaf. This is a bit broad leaf. Here parallel venation takes place, and here reticulate venation takes place. Okay, and now here in the monocots, especially coming to the monocots, if you see the petals, as we have studied now, three petals, 5 petals and all, no. So this particular leaf will have the 3 petals. So that will be the multiples of 3. They will have the multiples of 3. The petals will be arranged like this, 3, and after that 3, and again on that there will be 3, like this, all 3, 3, 3. So now this can be called as the multiples of 3. So here you can see the 3 petals, right? 3 petals, and also the otherwise you can say multiples of 3 petals here. And when you come to the dicots and all, you can see the multiples of 5 petals, 1, 2, 3, 4 and 5, and again 1, 2, 3, 4 and 5. Like this, you can see multiples of 5 petals here, and whereas in the monocots you can see multiples of 3 petals here. So this is how you can write the differences of the monocots and the dicots. Is it clear, children? So what is the major difference? Here it will have only one embryonic leaf. It will have 2 embryonic leaves. Okay, and here because it is there is only one embryonic leaf, we call it as the monocotyledon. Here there are 2 embryonic leaves. They are called as the dicotyledons. They are not the true leaves, but they comes out at the time of germination of the seed, and also they will supply the nutrients to the leaves until they are able to, or they become capable of performing the photosynthesis process. And here you can see in the monocots you can see the parallel venation, and whereas in the dicots you can see the reticulate venation. And also here you can see the flowers, the petals are in the multiples of 3, and here you can see the petals are in the multiples of 5. Okay, and one more difference is there. Okay, now you know if you see the vascular bundles, that is what we have studied about the xylem and phloem. Okay, the xylem and phloem usually are present at the edges. In the monocots and all, they are present at the edges, and they are scattered. They are not in a right particular perfect shape. Okay, so they are scattered at the edge of the stems like this. Okay, and whereas when you come to the dicots, they are in the exact ring-like form. Okay, so they maintain a ring-like structure here, and they are perfect in shape, but whereas here they are scattered. They are present in the scattered way near the edges of the stem. Okay, and here the xylem and phloem are arranged in a ring-like structure. So these are the 5 differences which you can see in the monocots and the dicots. So hope children you understood the monocots and dicots. Okay, children, right? So shall we go to the okay, children, now we'll go ahead with the third activity, activity number 3, observation of seeds. Observation of seeds. Again, here you have to collect some of the seeds from the different plants and all. Okay, you can collect the seeds of green gram, red gram, Bengal gram, wheat, paddy, groundnut, maize. Okay, all of them, and soak them for a day. Okay, and observe them very, very carefully. Okay, if you take a maize seed, okay, the next day morning, and try to press it, okay, you know what happens? A white color part will come out, isn't it? And that's nothing but the embryo. Okay, and check whether the same thing happens with the other seeds also or not. If they come out, okay, that is monocot. No, if it breaks into two, then it is dicot. Okay, maize, it has only a single coat around it, and the inner part, the white color part, which is nothing but the embryo. Okay, and then if we take the other seeds and all, they will break into two, and which are going to grow as a dicotyledon, that is two embryonic leaves. Okay, so that is how you can easily check whether the seeds are monocots or the dicots. The plants are monocot plants or the dicot plants and all. Okay, now here the same thing what we'll observe here is when you observing the seeds, otherwise you can say observing the cotyledons in the seeds. Now take some plants, name of the plant you can write, color of the seed you can write, and shape of the size also you can write, whether it is round, whether it is triangle, whether it is elliptical, okay, whether it is long. Okay, so whatever the different shapes you see in the seeds and all, you can write the seeds and number of cotyledons. So now we have seen the way how to check the cotyledons. So you can say what kind of, how many cotyledons are present there, you can write it. So other characters, so based on all these things you can write them whether they are the monocot plants or the dicot plants, and also you can write some other features which you can find in these plants. Okay, so that is what you can see here. Here we have a table here again. In the table there are 7 different seeds which are given here, like green gram, red gram, Bengal gram, wheat, paddy, groundnut and maize. Okay, even if you check the colors, there are different colors: green, red, black, gray, yellow, pink, yellow. And shape, size and seeds leaves, you can say they are round, small, elliptical, round, triangle. So these are the different kinds of shapes you can find in the seeds, right? And then number of cotyledons, you can see some of them have the 2 cotyledons and some of them have the 1 cotyledon. And then what are the characters observed based on all this? So few of them are the dicot plants and few of them are the monocot plants. So this is how you can observe the seeds. Okay. So now what we'll try to do is we'll also try to observe the monocot plants and also the dicot plants. What we'll do is we'll observe the monocot plants and as well as the dicot plants here. Okay, so as we have already studied here, there are different characters in the monocots and the dicots. So already we have discussed about the differences between the monocots and the dicots, and one point, like you just try to remember, in the monocots, it is always a fibrous root system. The kind of root system you can find is the fibrous root system, and whereas when you come to the dicots, you can find the tap root system. Okay, this is the one which will add to the differences in the monocots and the dicots. Okay, so now we will try to see the characters of the monocot plants and the dicot plants. So children, now here what we will try to do is we will try to compare the dicots and the monocots. So that is what are the differences which we have already seen, right? So what are the differences we have seen in the monocots and dicots? Okay, we have seen around 5 differences, right? So what's the first one? Monocots will have only 1 cotyledon, dicots will have 2 cotyledons. Monocots will have the parallel venation, and whereas dicots will have the reticulate venation. Here the leaves will be long and slender, here the leaves will be a bit broad. Okay, and then you can see petals are in multiples of 3, and whereas in the dicots petals are in the multiples of 5. Here the xylem and phloem, the vascular bundles, are scattered and they are present in the edge of the stem, whereas in the dicots they are present in the ring-like structure, isn't it? And the last one is we can see the fibrous root system in the monocots, and in the dicots what you can see is the taproot system. So now what you do is you go and select 4 or 5 or 6 plants like this, okay, which have the characters of the monocots and dicots, and then write the name of the plant. Okay, write the venation of the leaf, is it a parallel or reticulate venation, and then write the number of cotyledons, and then write the root system. So what kind of root system is present over there? Is it a fibrous root system or a taproot system. So we can easily come to a conclusion that, okay, whether this is a monocot plant or whether it is a dicot plant. So if it is a monocot plant, what are the characters it have? If it is a dicot plant, what are the characters it has? So by doing all these activities, all these 4-5 activities, what the conclusion we come to is, in plants itself we can see variety, right? A complete diversity. You can see different colors of the seeds, different shape of the seeds, different venations, different margins, different monocot and dicot systems. Okay, so and even the size of the leaf or the shape of the leaf, the nodal length, the placement of the nodes, everything will differ. So by this we come to the end of the diversity in plants. So by these 4 to 5 activities, what we can learn is there is a great diversity in the plants. So that is how all these experiments will help us to know the diversity in the plants. So children, so hope you all understood today's topic, and we'll see you tomorrow. And if possible, maximum do this experiments at home. Try to check all the leaves in your or plants in your gardens, okay, in your surroundings. Get them and check whether they are monocots or the dicots. Check the seeds whether they are monocots or the dicots, okay, whether they belong to they are monocotyledons or the dicotyledons. See their venation. See if it's a monocot, see whether it has a fibrous root system or not. If it's a dicot, see whether it is a taproot system or not. Okay, if it is a monocot, check whether the leaf is having the parallel venation. If it is a dicot, it is having a reticulate venation. So check all these things, right? So if you check everything on your own, you can find a lot of things on your own. So this is how we come to the end of the session, which is the diversity in plants. Hope you all understood. Okay children, so see you tomorrow with a new topic, diversity in animals. Until then, have a great day. Take care.